Thank you, sir. And I thank all of you for sitting through the last talk. And I am Sunil Arora. I come from India, a very small town, Chandigarh, which is north of Delhi. And I bring greetings from that small town, which is also called City Beautiful because it's a newly made town and has got it's an education hub in India where we have a university, medical school, medical center, and many other institutions. So I'm going to talk about Leishmania parasite, and the disease, if you don't know, is a wide spectrum disease. It has a wide spectrum, clinical spectrum, starting with the localized self-healing lesions. You have a killing, which is lethal visceral lesions also. As the clinical spectrum varies, we have the immune spectrum also, which varies. If we look at the self-healing lesions, you have very few parasites, but the cell-mediated immune response is very high in these patients as the lesions they heal. Very low antibodies. Whereas on the other side, when you have the visceral leishmaniasis, which is more lethal kind, you have many parasites, but the cell-mediated immune response is almost missing in them. And you have high antibodies, high level of antibodies, that's mainly because of the polyclonal B cell activation, which shows that the cell-mediated immune response is somehow protective, whereas the antibody response is not very protective. There are chemotherapeutic agents available which take care of this, but they are limited, toxic in nature, they are very expensive, a long duration of treatment is required. There is development of resistance which has been, evidences, and there are cases of relapse which have been seen. So that means that the treatment with chemotherapy is not 100%. There are always parasites which remain in the nature. Regarding vaccine against Leishmania, there are no vaccines as on today, and the development of drug resistance in chemotherapy emphasizes that there is a need for a vaccine. And there are cases where immunity is lifelong after the cure of Leishmaniasis, which emphasizes that the approach is feasible at least because a person who develops immunity against further infection. There are subunit vaccines which have been tried. They're mostly antibody-based, and we know that the protective immunity is more of cell-mediated immune response rather than the antibody rate. So an antigen which is defined can elicit a Th1 type of specific response may induce protective response. There are so many antigens which have been tried with a limited success, and there are some more list, but there have been very limited success. Nothing has come out as a positive response. There is at least a couple of them have reached clinical trials, which is underway now, but the protective response has not been very, very successful. So there are research gaps, which shows that the basic knowledge of the complex interplay between the parasite and the host is not yet well understood. There is development of new therapeutic regimes are required. Then studies on the disease pathophysiology, genetics, and host immunology is again required, and the characteristics of the parasite components may also be helpful in further treatment. So molecular immunology lab, which I am heading in postgraduate institute, we planned with the objective to look at the antigens which can stimulate specifically Th1 kind of immune response and be protective. In that endeavor, I know this is a very busy slide, but I'm trying to summarize work of so many papers in one slide, where we made a cDNA library and we screened it with the immune sera as well as Leishmania specific cell line which was generated, and we identified some clones which were expressing antigen which could induce very high level of interferon gamma production from the Leishmania specific cell line when used in vitro, and we selected 3 antigens, 3 recombinant clones which were novel in nature, and they were sequenced, and the the sequences were submitted to the gene data bank. We tried to look at the protective efficacy of two of these using a hamster model, and they were cloned in PQE vector to express high level of proteins, and we used some of the human compatible adjuvants, and these are the experimental groups where we use the combination of CpG-ODN with the antigen and also monophosphoryl lipid as the adjuvant. So these are two adjuvants which are known to induce cell-mediated immune response, so they will help the antigen to reduce, and we use two recombinant antigens. These are the immunization protocols. The primary immunization was followed by 2 boosters and challenge at the 42nd day with the 10 million AMST goats intravenously. And first parameter we checked was the survival rate of these animals, and we found that the combination of one of the recombinant antigen called F14 with MPL was able to induce survival for more than 12 weeks, while most of the animals in other groups, they started dying at that level. And further we checked that whether the spleen weight, because the parasite is intent to go and accumulate in the spleen, and the spleen enlarges, and this is where we found that this combination of this antigen with the MPL immunized animals, they resisted the increase in spleen weight, whereas all other animals had a splenomegaly. The parasite load was also very low in the spleen as well as liver in these immunized animals as compared to the other animals. The cell-mediated immune response was also induced as we look at the phagocytic index, and the killing index in the immunized animals was very high, as well as the nitric oxide production as well as the oxygen-free radicals were also induced very high. The delayed type hypersensitivity was also induced in these animals, and the cytokine response was also very good as the interferon gamma production was high and IL-10 production was low in these animals. So overall, we summarized these findings in one of the papers which published, and the Suvarcha, she explained that there was a partial protection. So it was about 67% protection. There was increased survival rate, lower splenic parasite burden, elevation of DTH response, and high interferon gamma. So all were good, but the protection was only in about 67%. There was another antigen which we cloned both used as a protein vaccine as well as DNA vaccine, just to look at if DNA vaccine can work better than the protein, and we cloned the one full complete open reading frame of this gene, which was cloned in PQE vector, and we made the protein vaccine as well as we used the PVAX plasmid base to use it as a naked DNA vaccine. And the animals were immunized with protein vaccine in the same schedule with the immunization followed by 2 boosters and the challenge, and also DNA vaccine, which was used as a PVAX only plasmid and the cloned one, and the 2 boosters of this, the prime and the boost effect, and then all the animals were challenged with the AMST codes. The protein vaccine was not very successful in inducing the protection, whereas the DNA vaccine, when we used in the prime boost mode, was found to reduce the parasite burden, reduce the spleen weight, and also induce the cell-mediated immune response, and the parasite load was very low in these animals which were immunized with this. The excretion of TH1 type of cytokines, the interferon gamma and IL-12 was high, whereas TH2 type of cytokines was low. And the animals, the immunohistochemistry showed that they had mature granuloma, which is a sign of protection in these animals, and we summarized this in one of the papers, which showed that there was a significant reduction in the parasite burden, which went up to 75%, where increased expression of TH1 type cytokines, formation of mature granulomas, and which all showed that the vaccine we generated, DNA vaccine, was working but not 100%. So the interim conclusions, they say that the recombinant vaccines were not 100% efficacious, while chemotherapy is toxic. There are now evidences with the incomplete cure with the chemotherapy. So, and we know that the Leishmania down-modulates the macrophage functions. So an immunomodulator, if can be used, which can activate the macrophage functions, can actually help in the early clearance. So we chose one of the recombinant antigens which we used as a prophylactic vaccine because they showed evidence that they boost the cell-mediated immune response and they activated the macrophages. So we planned another strategy where we found that the intracellular parasite clearance can be increased if we use one of the recombinant antigen as adjunct candidate along with the chemotherapy. So whether it can reduce the toxicity of the drug and also activate the macrophages' effector functions. So the approach used was that we used the macrophages, infected them with the Leishmania, and then try to activate that ex vivo with this antigen and see if there is any assessment of the enhancement of the macrophage effector functions and monitor the intracellular clearance also. So this antigen was able to induce the nitric oxide production at 24 hours. And when we used in the these mouse peritoneal exudate cells as well as one of the cell line which is macrophage-like cell line, in both when we use this recombinant antigen, it will induce the nitric oxide production to a very high level alone as well as in the infected conditions. So we could check the intracellular expression of reactive nitric oxide using diamino fluorescein dye, which changes to triazole, and it is fluorescence, and you can use flowcytometry, and it shows that intracellular nitric oxide production as well as intracellular reactive oxygen species were also induced very high when we use this recombinant antigen. This also induced the expression of inducible nitric oxide synthase as well as TNF-α gene in the activated form. And it also increased the phagocytic index as well as the killing index of these animals as it was used. We looked at the Staphylococcus aureus infection and then killing and phagocytosis. This antigen also induced right kind of cytokines where it induces the TNF-α production, MCP, IL-6, and reduced the IL-10 production on ex vivo production. Since it was expressed in E. coli, so we checked whether it was contaminated with endotoxin or not. So we found that R6 was not endotoxin contaminated because the level of endotoxin was way below the threshold level of this. We checked the intracellular clearance by looking at the PKH67 labeled parasites, and we could use the flow cytometry and showed that when we use the activation by RA6, the level of fluorescence decreased, which means the intracellular parasites were getting cleared as we use this antigen in the infected parasites. So finally we planned another experiment, which was that we took the Leishmania parasite, infected the macrophages, and then the infected macrophages were either cultured in medium only or the antigen or the miltefosine, which is the anti-leishmanial drug, or the combination of miltefosine and R6, just to see whether there is any adjunct effect of this recombinant antigen which can reduce the dose of this parasite. Look at this combination. These are all, the infected only untreated ones, treated with recombinant antigen, treated with miltefosine, which is the anti-leishmanial drug, and the combination. And we found that the parasite clearance was very, very effective when we use the combination of these two. And the dose of drug was almost half the dose of drug, and the parasite clearance could be achieved much earlier as compared to the clinical level of the drug which we use. Similarly, another anti-leishmanial drug, which is ambisone, which again a very toxic, but when we used it in combination, the parasite clearance was achieved much earlier and with a subclinical dose also of this. So we summarize that the Leishmania, when it infects the macrophages, it down-modulates the functions of this macrophage and then survive inside it, and it multiplies, which causes the disease progression. If we use this recombinant antigen as a immunomodulator, it leads to clearance of intracellular parasite to some extent because it up-regulate the functions of effector functions of macrophage. So there is immune modulation effect. The drugs can also clear the parasite, but not 100%. When we use the combination of this miltefosine and recombinant antigen, there was early clearance. There was upregulation of all these macrophage effector functions, and this could be achieved at a much lower dose of the drug. So the translational prospect is that we can achieve an early and complete clearance of intracellular investigotes when we use the recombinant antigen along with the anti-leishmanial drug, which will reduce the duration of treatment, which will reduce the dose of the toxic drug, and it makes a promising candidate as an adjunct therapy. So the Leishmania, the evidence is that Leishmania recombinant antigen modulates macrophage effector functions, synergizing anti-leishmanial effects of miltefosine to facilitate early clearance of the intracellular parasites. So I would like to acknowledge the contribution of my students. Nirbhay Paul, he was the one who initially screened the cDNA library and identified the clones. She worked on the protein vaccination model. He worked on the DNA vaccination model. And Arora is now working on the combination and immunomodulatory effect of this antigen. And I acknowledge the funding by DBT and ICMR, and this is my institute. And finally, I thank you for your kind attention.
Very interesting. Now, such a small protein and single protein out of hundreds of proteins can have a protective immune responses, isn't it? This F14. So what did you put into your DNA for DNA vaccine? Which genes are in there?
Initially, I showed you 3 clones. Out of these, 2 were used as protein. Third was
no, in your DNA vaccine. So which but plasmid containing which genes?
The gene which we cloned.
Yes, those 3.
One of those.
I see. So single gene expression can make a protective immune response.
Yes.
Wow. Out of hundreds of proteins of Leishmania, right? Any other questions? Yes.
So recombinant B6 used is for induced nitric oxide production. Do you think this will be benefit effect for Leishmania or not? Because nitric oxide can suppress T cell response. So if you induce nitric oxide in other macrophage which is not phagocytic Leishmania, it might suppress T cell response. So have you considered about this?
Leishmania by nature, it suppresses the oxidative burst. So oxidative burst will require the reactive oxygen species and nitric oxide also, which is a part of killing mechanism of macrophage, right?
In the meantime, so if nitric oxide can also suppress T cell response.
No. That's okay. But the intracellular production of nitric oxide is very important to kill the intracellular parasite. Yeah. So it doesn't matter if, actually, why do we need Th1 response? We need Th1 response for interferon gamma, which ultimately activates the macrophage for better production of reactive oxygen species and nitric oxide. And these 2 are very important for killing the intracellular parasite. It doesn't matter if the T cell response is low, still the macrophage is activated, it will clear the parasite. So macrophage is the host cell for parasite. T cell is not the host cell for macrophage. We don't care if it works or not. So basically we are working always to induce the T cell immune response. ultimately that activates the macrophage only. So if we are able to activate the macrophage by other mechanism, it will help in clearing the parasite.
So the idea for the vaccination for anti-leishmania is to induce Th1 response. Yeah. So have you considered using like L12, L23 to induce Th1 response combined with your antigen or your DNA vaccination?
See, there are adjuvants. There are some adjuvants which induce Th1 kind of immune response. We already tried those, you know. We tried CpG-ODN. We tried the monophosphoryl lipid. We even tried some PLG particles. They all induce Th1 type of immune response. But we realize after so much of vaccination protocols that they induce protective immune response, but it is never 100%. You don't reach a sterile immunity. That's why the vaccine is evaded. You know, till now we don't have a vaccine.
I suggest that you two get together over the beer and then discuss about this problems. And I appreciate all the speakers. And also, you may wonder why I'm chairing this, because Dr. Hannon did not show up early in the session. So, unfortunately, I volunteered it, and then now I'm stuck here until 7 o'clock this evening. But anyway.
I thought maybe two of us will be stuck.
Anyway, and I appreciate you being so patient and then staying here until the last minute. And also, Dr. Arora, here is the certificate of appreciation. And thank you for your presentation. And have a good evening.